Listcroft and Harmon Deep II with the help of an amazing graduate student, Julie. And the work concerns sexual selection's effect on purging deleterious mutations, and in particular, the effects of simple versus complex mating environments on this. So I'm going to start with a little bit of background. So in the Origin of Species, Darwin suggested that selection arising from variation in reproductive success, that is sexual selection, should, in his words, give its aid, I would say align with, selection arising from variance in other components of fitness. Fecundity, viability, I'll call that natural selection for short. And this is because the highest quality, best adapted males are going to sire the most offspring. And if we were to restate that in a bit more modern lingo, I'm borrowing from the writings of Locke, Rowe, David Houle, Mike Whitlock, and Neil Agarwal, we'd say something like, if reproductive success is dependent upon the condition of an individual, their health or their vigor, then sexual selection will align with natural selection. And because much of the genome likely contributes to how healthy and vigorous an individual's condition, there should generally be genome-wide sexual selection favoring alleles of high non-sexual fitness. In other words, sexual selection should promote adaptation, the fixation of beneficial alleles, and it should help in the purging of deleterious alleles. Now, there's been several empirical tests of this. They've taken a couple different approaches. You can manipulate the opportunity for sexual selection, create a monogamy treatment with no sexual selection, and a treatment where you have a mixture of multiple males and females, so you have intra and intersexual selection. And then you can measure the consequences for population mean fitness across one or more generations. Or you can track the frequency of individual alleles, beneficial or deleterious, through time under those treatments. Or an alternate approach has been to measure the effects on male reproductive success of alleles that are deleterious to viability or fecundity. That, all right, if it lowers viability, does it also lower male reproductive success, which would be alignment. Now, there haven't been a huge number of these studies, but there's been enough to conclude that the results are quite variable. So there's a couple studies that show a benefit of sexual selection, that is an alignment with natural selection. There's been a couple that show no net effect of sexual selection. And there's been a couple, including two recent ones in my lab, that show that sexual selection seems to hamper natural selection. And so, I think the more interesting question is what's going on here? Why are the results so variable? And in particular, why might sexual selection not align? Why might it be antagonistic to natural selection? And there's a couple of reasons why that may be the case. First off, by generating variance in male reproductive success, or it relies on that, sexual selection reduces the effective population size, increases drift, and may therefore hamper natural selection. Secondly, sexual selection is a common reason why selection differs between the sexes, that is, selection is sexually antagonistic. And this generates sexual conflict, which can reduce female mean fitness and possibly population mean fitness. And this can come about through intralocus conflict, which creates a gender load, and through interlocus sexual conflict, which drives the evolution of traits in males that can be harmful to females. But these two processes on their own, while they may lower population mean fitness, they shouldn't hamper the purging of deleterious mutations across the genome or fixation of beneficial ones. But as Tristan Long and Bill Rice and colleagues pointed out in a 2006 paper in PLOS Biology, in species with mating systems that feature male mate preferences and male-induced harm, that is from sexual conflict, you can get a hampering effect. And that's because the male mate preferences cause the male harm to be disproportionately directed towards the otherwise best females, and that reduces their fitness, decreasing the variance in fitness, and thus hampering the response to selection. And graphically, that looks something like this. So imagine a female here carries a high fitness and beneficial allele in a particular locus. She's big, plump, fecund. This female over here has lower fitness allele at that locus, deleterious mutation perhaps. She's smaller, less fecund, less vigorous. If you assign each of those females a random male, so monogamous conditions, she's going to leave more offspring because of this higher fitness allele at that locus than she is. But now if you mix multiple males and females together, the males focus their courtship on this larger, plumper, more attractive female. That male sexual attention is harmful. It reduces her fecundity, and it may reduce the advantage of carrying that mutation, that allele, that high fitness, otherwise high fitness allele. It may entirely eliminate the advantage, or it could even reverse it if the cost of this increased sexual attention more than offsets the benefit of that allele directly to her fecundity. And as I said, we've done two recent studies out of my lab, one by a former PhD student, Devin Arbuthnot, which is looking at the purging of deleterious mutations. And another one that just came out last week, actually, in collaboration with Steve Chenoweth's lab at the University of Queensland, which was looking at adaptation to novel abiotic environments. Both of these found a net cost of sexual selection, that is the opposite result to alignment. And follow-up studies implied this cost of sexual attractiveness as the explanation. So I've been on sabbatical at the University of Toronto for a little while, and Anil and I spent a fair amount of time discussing these past studies because we were both quite surprised at the results of this previous work out of my lab that showed a net cost of sexual selection. And in particular, we were really surprised at the apparent strength of this cost of sexual attractiveness sort of effect. And our discussions led us to a consideration of the mating environments used in not just our study, but almost all the studies that have been done in this system, because the vast majority are insect and, in fact, Drosophila based. And the mating environments look like this. It's a vial or a bottle. You're familiar with the Drosophila environment. These are spatially restricted, small, and structurally totally, there's no complexity there whatever. It's all a simple environment. Just a bit of food, a bit of empty space, and individuals are usually kept at reasonably high density in those. And we hypothesized that environments like this may predispose you to finding a net cost of sexual selection for two reasons. One of which, it reduces the benefit. And the benefit, remember, comes about through condition dependence of male reproductive success. In male reproductive success, environments like this may just be less condition dependent. Certain components of male sexual fitness don't matter in an environment like this. For instance, searching out, locating a female. It's kind of easy when they're locked up in front of your face. Males can't express territorial behavior, which if you keep them in a larger environment, lower density, they will. And so even a relatively low quality male may do pretty well in terms of reproductive success when faced with a bunch of females who can't get away. So reduced benefit. Secondly, the cost, remember, comes about through this cost of sexual attractiveness, and that relies on male mate preferences and male harm. Well, the opportunity for a male to express a mate preference may be greatly enhanced here because, again, he's confined to the whole array of females he can choose among who can't get away. And secondly, the male harm may be increased in an absolute sense because, again, she can't escape harm when she doesn't want to. Just watch some Drosophila in the wild, you notice that females come and go from mating arenas, possibly to avoid male harassment. And so, more generally, we're hypothesizing that the population genetic consequences of sexual selection for adaptation and reducing mutation load may be environment dependent, and we may be looking at a particularly weird environment here. And so that's what we wanted to explore. So we took 4 of the mutations that we used in the previous study. These are recessive mutations, brown, sepia, plexus, and white. 2 mating treatments, a simple and a complex. These are Ziploc containers, about this big, a little bit bigger than the standard Drosophila vial. We had a simple one with just a petri dish of food, and a complex one. The food now is in these spatial patches, little Dixie cups, and some of them, there's an additional barrier across the food surface, such that a female that's on one side can't be seen by a male on the other side and harassed. And then there were these pipe cleaners that stuck down from the lid, so the lid flips upside down on top of this, which provided perches for the females and places to hide from males. And so for each mutation, we created 6 replicate populations with a mutant allele frequency of 0.65. 3 of them evolved in a simple environment, 3 in a complex. Every generation, a bunch of females and males went in their mating environment for 6 days. Females are then removed and lay eggs for the next generation. So natural selection is present here all the time, as is sexual selection, but the environment in which mating is taking place differs in this way. 150 females per generation laid eggs, and we track the frequency of the mutants, both by the visible homozygotes, because you can just tell them by eye, and then we use test processes to actually estimate allele frequencies. So just go through the results in turn for each mutation. This is brown. So we get generations across the x-axis here, mutant frequency on the y-axis. The solid lines are the genotype frequency of mutant homozygotes, and the dotted line is the mutant allele frequency from test crosses. Points are the mean for the 3 replicate populations with their standard error, and the blue is the complex environment, and red is the simple. So for 2 of the mutations, brown being one of them, there was no effect of the mating environment. You can see there's no difference in allele frequency there. It's a t-test for the difference, two-sample t-test for the difference in allele frequency between treatments, treating populations as replicates, and there's just no effect here, and you can see that the homozygote mutant frequency just kind of bounces around, very similar in the 2 environments. Same thing with sepia. There's no difference for this mutation. They had a little bit of trouble with the genotype frequencies at the start with some death, but the allele frequencies varied only a tiny bit. But it's pretty clear that across generations here, there's no sign of any treatment effect there. However, for the other 2 mutations, white being one of them, we had a significant treatment effect, that is, the mating environment, and in both cases, the mutation declined in frequency. It was purged faster in the complex than the simple environment. So you can see that after only 6 generations, white was significantly lower in frequency in the complex and the simple environment. You can see that in the mutant homozygote frequencies as well. And you see the same thing for plexus. And you can actually see the difference here building through time with plexus. We ran this one a little bit longer. Their honors project was done, but they were interested in keeping this going a bit. And so, for 2 of 4 mutations were purged faster when the mating environment just included a little bit of ecological realism to it. Just a little bit of spatial structure, that's it. And it suggests that sexual selection is better aligning with natural selection in a slightly more realistic environment. Although, we don't have a monogamy treatment here, so we don't know if that is, we know from Devon's previous work that sexual selection was costly for these mutations. We don't know if this change we're seeing is enough to actually turn it over to being beneficial now. That's what we'll have to address in future studies. But certainly it seems to be aligning better. Either the cost has been reduced or it's actually beneficial now. And whether this is due to the effects of the mating environment on the condition dependence of male mating success, or this reducing the cost of sexual attractiveness, or both, we don't know. We're doing some follow-up studies to look at that. But it suggests that the costs or benefits, it implies that the costs and benefits of sexual selection depend on the particular environment. So the next step, some ongoing work. Julie, in my lab, is trying to quantify the effects of space. We only looked at temporal, we only looked at structural complexity here, environmental complexity, and previous life history, because that can affect the strength of male harm on both the cost of sexual attractiveness and the condition dependence of male mating success. And then in collaboration with Neil, we've got 2 graduate students, one who's doing a similar experiment to this one, but using a broader set of mutations, the exelixis lines, and they lack visible phenotypic markers, which is great. And then Lee here is looking at adaptation to a novel environment, adding structural complexity. So thanks for your attention, and thanks to Neil for helping develop these ideas, and you for your attention.